So I'm Oliver Howes, and I'm going to be talking about the neurobiology of risk and resilience for psychosis and novel approaches to target the dopamine system in particular. I want to start by just acknowledging lots of people who've helped with the work that I'm going to talk about, and particularly for the two studies that I'm going to present, Maria Rogdaki, who you can see on the left of the screen, and Michelle Coquinou, who's on the right. Both of these took a lead in the separate studies. And I'm going to talk about two studies today. I'm going to start with a clinical study looking at genetic risk and resilience factors and the neurobiology underlying these, and then a preclinical study that looks at potential approaches to target that neurobiology, potentially to treat psychosis, but also even to prevent psychosis and support resilience. So in terms of the neurobiology, I'm going to talk a lot about the dopamine system. And of course, the dopamine hypothesis of psychosis and schizophrenia has been around for many years. It has evolved quite a lot since that early publication in the 1970s by Meltzer and Stahl. And now we know much more about not just the nature of the in vivo changes in the dopamine system in psychosis, but also the various environmental and genetic factors and how they might act on the dopamine system. And here you see a summary from a recent paper that discusses this. And of course, on the left, there are lots of environmental risk factors for psychosis, but also there are many genetic risk factors for psychosis. And I'm going to be talking about a particular genetic risk factor and the reciprocal resilience factor, and whether this might actually be acting on the dopamine system. And another way that the dopamine hypothesis has advanced is that we now know that in vivo there are alterations, particularly in the synthesis capacity and storage capacity and release of dopamine in the striatum, and that this is linked to the psychotic symptoms that you see in schizophrenia. And how do we know this? Well, we've been fortunate that there are lots of PET tracers that allow you to measure different aspects of the dopamine system. And these are summarized here from a review by Jody Weinstein a few years ago. And I'm going to be particularly talking about PET imaging measures of dopamine synthesis and storage capacity, which you can image in vivo in patients using radio-labeled L-DOPA, which is converted into dopamine, and that you can image that process in patients. And this has been done extensively in people with schizophrenia. Now, over 20 studies showing that there is a large effect size increase in this uptake and conversion of radio-labeled L-DOPA into fluorodopa and its storage in vesicles in dopamine neurons. So this is something that's seen in schizophrenia and is linked to the psychotic symptoms. It's something that we've also seen in people at clinical high risk of schizophrenia. But the question that we wanted to ask is whether this is something that could be mediating the genetic risk for schizophrenia and could be part of the underlying neurobiology that links genetics of schizophrenia with the development of the symptoms. Now, we focused on what is arguably the single biggest risk factor for schizophrenia, and that is a 4 megabyte deletion on chromosome 22 at 11.2. This is called the deletion, 22q11 deletion. And you can see that deletion involves quite a few different genes as shown here, including, you can see one for COMT, C-O-M-T, which is involved in the metabolism of dopamine. So there are reasons to think that this deletion might affect the dopamine system. And it is a major risk factor for schizophrenia. As I said, people that carry this deletion have about a 40-fold increased risk of developing the illness, and over 40% of them develop a psychotic or schizophreniform illness by their mid-20s. So it really is a major genetic risk factor. What's really interesting, so, is that the duplication of the DNA at this region, and 22q, has been found to protect against schizophrenia. So again, you get a copy number duplication of about 3 megabytes that protects against schizophrenia, reducing the risk by quite a considerable amount. Now, this was the first publication on this a few years ago. Since then, there have been a number of other publications. It's been replicated in some, and in some of the others, it hasn't been replicated. So this is not as well established as a resilience factor against schizophrenia as the deletion is a risk factor for schizophrenia. But nevertheless, there's a few converging findings that suggest it does protect against the illness. So this led us to wonder whether these genetic risk factors might be acting through the dopamine system. So we wanted to first see whether the people who are carriers of the 22q11 deletion showed elevated striatal dopamine synthesis and storage capacity, which is what we see in people with schizophrenia, but also in people at clinical high risk of the illness as well, and whether this might be linked to subclinical symptoms and potentially the development of psychosis. But we also wanted to see whether this reciprocal duplication copy number variant, so having extra copies of that same amount of DNA that is deleted in the deletion carriers, whether that actually could be protective through reducing dopamine synthesis capacity and storage, and that might be what the mechanism why it leads to lower risk of psychosis. So this is the clinical study that Maria Rogdaki carried out with others in our group, and this is the sample characteristics of the people that she scanned using that fluorodopa PET imaging technique that I talked about. So she was able to scan over 20 deletion carriers, and the important point to note about them is none of them had experienced a psychotic illness before being scanned. So they were generally quite young, and we excluded anyone who'd had a frank psychotic illness or had been treated with any antipsychotics. And this is because we wanted to look at people with a confirmed genetic risk for schizophrenia who had not yet experienced the illness. And they were compared with duplication carriers, and again, they hadn't had any psychotic illness before being scanned, but as I said, they are at a reduced risk of psychosis anyway. And then 26 matched controls. And here you see the gender characteristics of the sample, but also the symptom ratings. And the CALMS is a rating scale for subclinical psychotic-like symptoms. And you see the total scores there, and also the score of the positive or specifically the psychotic-like symptoms at a subclinical level, but still present in the deletion carriers, and also to some degree in the duplication carriers. It's also worth noting that both the deletion and duplication carriers also had low levels of depressive and anxiety symptoms as well. And in fact, the duplication carriers had significantly higher depression ratings than the deletion carriers. So firstly, what is the dopamine synthesis and storage capacity in these groups? So here you see on the vertical axis the rate constant for the uptake and storage, synthesis and storage of fluorodopamine in the striata of the duplication carriers, then the healthy controls, and then the deletion carriers. And you can see that the deletion carriers show a significant increase relative to both the healthy controls and the duplication carriers. And in fact, that there is a linear relationship in dopamine synthesis capacity across these three groups that is significant as well. So that was in the whole striatum. We also looked at subdivisions of the striatum, and we saw the same pattern across all the subdivisions. This was really influenced by recent findings that suggest that the major dopamine dysregulation in schizophrenia is particularly in the associative striatum. And this is something we've seen in schizophrenia and also in bipolar psychosis as well. But you see here in these genetic risk carriers, the deletion carriers in blue, they seem to show this elevation in dopamine synthesis and storage capacity across all the whole striatum, across all the subdivisions. Next, we then looked at the relationship between striatal dopamine synthesis capacity and symptom severity in the groups of genetic risk and resilience carriers. So here you see the correlation between the CALM's positive symptom score and that striatal dopamine synthesis capacity in the deletion carriers shown in the blue triangles and in the duplication carriers in the red triangles. And you see that there is a positive relationship, and that individuals that showed higher striatal dopamine synthesis capacity had higher PANSS positive ratings or more severe subclinical psychotic symptoms. And you see that overall there was lower striatal dopamine synthesis capacity and symptom ratings in the duplication carriers, who are those that seem to be at lower risk of psychosis. So this is again consistent with the model that we were testing. Importantly, there was no relationship between dopamine synthesis capacity and either depression or anxiety scores. So again, it seems to be specific to psychosis or psychotic-like symptoms. We also measured brain glutamate levels in the deletion carriers using magnetic resonance spectroscopy and MR imaging technique. We found no differences in the deletion carriers compared to controls in these glutamate measures across a number of brain regions. So this again seems to be an aspect of neurochemistry that is specific to the dopamine system. It's certainly not seen with the glutamate system in the deletion carriers. So in terms of the model that we had, we found evidence that the deletion genetic high risk group showed increased dopamine synthesis and storage capacity prior to developing any psychotic illness, although we know that about 40% of them will go on to develop schizophrenia or a schizophrenia-like illness in the next few years, and that this was related to the severity of their subclinical symptoms. And we also found evidence that the genetic low risk or resilience group had lower striatal dopamine synthesis capacity, and that this was linked to symptoms as well. So this is evidence that some of the genetics of schizophrenia are acting on the dopamine system. We've also looked at some other genetic risk factors for schizophrenia recently, and DISC and the arsenic metabolic route are two of these that we've also found that are linked to alterations in striatal dopamine synthesis capacity. So this is suggesting that this is a potential neurobiological mechanism that might mediate genetic risk for schizophrenia and also resilience. So that's the clinical study. This now brings me on to the preclinical study. And in this preclinical study, we wanted to ask the question, how might we act on this striatal dopamine abnormality to potentially help treat or prevent psychosis? And first off, we needed to develop a model that would reproduce the dopamine pathophysiology that we've seen in patients. And to do this, we treated mice with ketamine repeatedly over a number of days, and you see that we gave them the ketamine on 5 sequential days, then let the ketamine wash out of their system, and then scanned them with a PET scanner designed for rodents, but otherwise using exactly the same PET scanning technique that we use in patients to allow us to measure striatal dopamine synthesis capacity. Why did we use ketamine? Well, ketamine has been well recognized to induce schizophreniform symptoms in healthy volunteers and worsen symptoms in patients. So it has good face validity from that perspective as a potential model of psychosis. Here you see a meta-analysis summarizing all the studies that have examined the effect of ketamine and its induction of symptoms in healthy volunteers. And you see that this is a pretty reliable effect and there's a large effect size. So this was our model in mice, and the first data I want to show you is the evidence that this does reproduce the pathophysiology of psychosis. So in the first cohort of mice, we show a significant elevation in dopamine synthesis capacity in the striata of mice that had received this ketamine. Remember, it's after ketamine has been washed out of the system, so it's not an acute effect or an effect whilst the ketamine is on board. It's something that seems to last. And you can see that the effect size is pretty large, but in the same sort of ballpark as that of what we see in patients with psychosis. This has just been published in Molecular Psychiatry, just come out this month. So the next question was, well, what is the mechanism by which ketamine might be having this action? And in particular, does it require midbrain dopamine neuron firing? And we tested this using a chemogenetic or DREAD approach. And this uses receptors that are expressed in the particular neurons that you're interested in regulating that will only respond to an exogenous ligand so that you can inhibit their firing. So we use these HM4 DREAD receptors, selectively express them in dopamine neurons in the midbrain. And here you see the labeling with TH and M cherry showing that there is co-expression of these receptors in TH positive, that's dopamine neurons, in the midbrain. And we then gave an exogenous ligand that we know will affect the firing of these neurons when they express this HM4 receptor. And here you see when you give this exogenous ligand, CNO, you can inhibit the firing of those dopamine neurons. So this allows us to block the firing of the dopamine neurons in the midbrain and then test whether ketamine's action on striatal dopamine synthesis requires dopamine neurons to fire. So we used this approach. We had 3 groups, a control group, 1, another control group for just giving the exogenous ligands, CNO, and then a group that received ketamine treatment and saline, and then a group that received the ketamine treatment, but also this exogenous ligand to block the firing of dopamine neurons. And then all these animals then had a PET scan after the washout of the ketamine. And here are the results of this study. So firstly, you see that in this other cohort, this second cohort, ketamine again induces this elevation in striatal dopamine synthesis capacity relative to the control groups. And then you see that the application of the exogenous ligand, CNO, at the same time as ketamine blocks this effect. So by blocking the firing of the dopamine neurons, we've been able to block the effect of ketamine on striatal dopamine synthesis, storage, and capacity. So what does this tell us about how we might target dopamine regulation to prevent psychosis? Well, there are various targets here that we've recently reviewed in a paper that came out last year. But one of them at the bottom you see is the trace amine receptor. And this is expressed in dopamine neurons and regulates their firing. So here you see data from a study that used a trace amine receptor 1 agonist, the RO compound here, and you can see that it reduces dopamine neuron firing. And this effect is blocked when you give it in combination with a TAR1 antagonist, and also if you give it to TAR1 knockout animals, again you don't see any effect on dopamine neuron firing. And TAR1 agonists also reduce striatal dopamine levels as well in animals. So plenty of evidence that TAR1 agonists might regulate dopamine neuron firing, but we don't know whether this would actually reduce striatal dopamine synthesis and storage capacity. So this is what we wanted to test. And again, we used the same experimental model, this ketamine model, and used a TAAR1 agonist called SEP that is in development, and this was work in conjunction with Sanovian, and they are running some clinical studies on this at the moment as well. And here you see that, again, ketamine induced this striatal dopamine synthesis capacity, but if you gave it in conjunction with the TAAR1 agonist, SEP, that blocked the elevation in striatal dopamine synthesis capacity. So this provides evidence that activating TAAR1 to inhibit dopamine neurons and reduce their firing is a potential strategy to treat dopamine abnormalities that seem to underlie psychosis, and also prevent, even potentially prevent the onset of psychosis. So just to conclude what I've told you about today, firstly, I showed you evidence that people who are carriers of the 22q11 deletion, the single largest genetic risk factor for psychosis, have higher dopamine synthesis and storage capacity. And this is linked to the subclinical symptoms in these people, whilst 22q11 duplication carriers who seem to have a reduced risk of developing psychosis show lower dopamine synthesis and storage capacity. So this suggests that the risk and protective genetic factors are having opposite effects on the dopamine system, and that this could explain potentially how they're acting, and also identifies the dopamine system as a potential final common pathway for the neurobiology of the genetic risk for schizophrenia. I also showed you data that a ketamine model that we've developed is able to induce elevated dopamine synthesis and storage capacity that is exactly the same as we see in patients when we measure it using the same PET techniques, and also that a TAAR1 agonist can counter this effect, which identifies this as a potential preventative strategy. Thank you very much for your attention.